everyone. Today I am here with chapter 11, that is reproduction in plants. In the previous video you have read about, that means introduction of the reproduction, that is what is reproduction, the production of the new organisms from the existing ones, and what are the importance of reproduction. That means it is important for the existence of the life on the earth, to maintain the number of species, and the different modes of reproduction, that is asexual and sexual reproduction, and the difference between them. In this video, we will read about the different types of asexual reproduction, like binary fission, budding, fragmentation, spore formation, etc. And explanation with the help of diagram. So let's start first, that is binary fission. So bi term that is mainly used for two, and fission means breakdown. So binary fission, that is a type of asexual reproduction where a parent cell divides, resulting in two identical cells, each having the potential to grow to the size of the original cell. Binary fission that is common among the prokaryotes like bacteria, blue-green algae, etc. This is the diagram of the binary fission that takes place in amoeba also, in which first, that means nucleus divides, and after that cytoplasm divides, and one amoeba that give rise to two daughter amoeba. Next is budding. So budding that is also a type of asexual reproduction in which new organisms develops from an outgrowth, and that outgrowth is called bud due to the cell division, and at one particular site there is small bulb-like projection coming out from the yeast. Actually the budding that is most common in yeast, and in animals it takes place in hydra. That cell is called bud. Now let's see the diagram, then it will be clear. So this is a yeast cell. So first of all, a small outgrowth that comes outside, and after that, that means followed by division of a small amount of nucleoplasm, which moves towards that bud, which is followed by a small amount of cytoplasm, and then it may detach from the parent body and give rise to new yeast plant. Sometimes chain budding may also occur. Now the next method, that is fragmentation. Fragments, that means breaking into the small pieces. So fragmentation that is a type of asexual reproduction in which an organism simply breaks in individual pieces at maturity. These individual small pieces then grow to form a new organisms, and it is most common in Spirogyra. Spirogyra that is a multicellular filamentous algae, that means it is found in aquatic habitat. So now let's see the diagram of the Spirogyra, then it should be clear. So this is the diagram of a Spirogyra, in which that means its body is breaks into the small fragments, and each fragments will give rise to new Spirogyra. Now the next method, that is spore formation. The parent plant produce hundred of reproductive units called spores, and when the spores case of plant bursts, then the spores spread into the air. Under favorable condition, they germinate and produce new plants. So the examples of the spore formation that may take place, that is in Rhizopus, mucor, that they are the lower groups of plant fungi, and fern, mosses, etc. Actually what happens in fern and mosses, a special structure is present that is called sori, in which these numerous spores are present, and in bread mold they are present inside the sporangium. So it may be clear from the diagram. The diagram of the fungi bread mold, in which sporangium is present, in which numerous spores are present, and this is that means the diagram of the fern and mosses, in which sori is present. So this is sori in which numerous spores are present. Now the next method, that is tissue culture, and tissue culture that may occurs in plants and animals both. So plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues or organs under a sterile condition on a nutrient culture media of known composition. That means whatever the nutrient that is required for this, that is used. It is widely used to produce clones of a plant in a method known as micropropagation. And nowadays it is, that means, very much common because it is very useful also. So let's see the diagram. So this is the tissue sample is scraped from a parent plant, and after that it may transfer into the samples in which agar growth. That means agar, that is a nutrient medium, and after that means what happens, it may develops a tiny plantlets, and then these plantlets may grow into a small area, and after that it may transfer into the main field. So this is the method of the tissue culture. Whatever the topic that is discussed, that should be clear to you. In our next video we will read about the vegetative propagation, that means the types of vegetative propagation. Thank you.